All right, good morning. So now we're going to be looking at, or good afternoon, we're going to be looking at chapter 8 here. We're going to be looking at microbial genetics. All right, so genetics is the study of inheritance, the heredity of living things, how traits are passed on. We're going to look at things, or this covers things like transmission of biological property, which is those traits from the parent to an offspring, how those traits are expressed in an organism, which is the phenotype, the structure and function of the genetic material, how genetic material can change, such as a mutation, how we manipulate and change genetic material through genetic engineering. And so that's the field that's got different techniques by which we are able to manipulate DNA in the lab. So it's really pretty cool, has some pretty cool applications. So the genome is the sum of all of the genetic material that an organism has, all right, all of their genetic material. Most of the genome is going to exist in the form of the chromosome. When we think of chromosome, this is what we traditionally think of, all right, where it's coiled up, okay? Some can appear as plasmid or sometimes in certain organelles like mitochondria. Mitochondria have their own, and chloroplasts actually have their own DNA. We have plasmids, which is what you see over here on the right, which is a small circular, basically kind of extra piece of DNA that many bacteria have. Genomes of cells are going to be made mostly of DNA, but viruses can contain DNA or RNA. All right, so we want to look at some of the similarities and differences between eukaryote, which is what we are, eukaryotes, eukaryotic chromosomes, and bacterial chromosomes, because they aren't exactly the same. So eukaryotic chromosomes, you're going to have DNA wound around histone proteins. And so basically, and I may have mentioned this before, this is kind of like when you have Christmas lights and you want to wind them up and you have that little plastic tray that you wind them around. It gives you some structure to wind around. So when we're talking about it being wound around, so you would have it kind of coiled like this, and in the middle of these coils is this little protein here that they wind around. All right, so that's what helps it to supercoil and be smaller, or take up less space, I guess I should say. It's usually going to be diploid, meaning in pairs, or haploid, which would mean single. They can have a linear type appearance to them, even though they're coiled. Bacterial chromosomes are condensed into a packet with histone-like proteins. They can have 1, 2, or 3, and they're going to have these more, let's see if I can put this on here, is these circular chromosomes here. Okay, so these are going to be your plasmids. Even some fungi in the eukaryotic kingdom will have plasmids as well. So genes are the fundamental unit of heredity that's responsible for a given trait, say blonde hair, green eyes, brown eyes. And of course, that gets a little more complicated than that, but at the basic unit of one gene, one trait, if we want to talk about it that way, okay? It can also be called the site on the chromosome that provides information for a certain cell function. Traditionally, we've characterized this as a segment or part of DNA that has the code to make a protein or to make RNA. And of course, then we take that protein and we make everything else with it. All right. Basically, anyway. So genes are going to fall into these 3 categories. All right. So we're going to have structural genes, and these guys code for proteins. Okay. Then we're going to have genes that code for RNA, and this is going to be the machinery that we need to make those proteins. Right. We've got those ribosomal subunits that make proteins. All right. And then we have regulatory genes. This is what they used to call junk DNA back when I was in high school or junior high, just a few short years ago, really long time ago. And so they called it junk DNA because they didn't know what it did, which never really actually made sense to me, because I always thought, why would we have something that we don't need? It's got to have a purpose, and it does. It has a purpose. It's basically like on and off switches, regulatory genes that help to control the expression of those traits. Genes are not always expressed, okay? So the sum of all of these 3 types of genes is what we call the genotype. That is the sum of all the genes that an organism contains, okay? Phenotype, and the best way to help remember this is that is going to be the physical pH phenotype, physical pH expression of those traits. So I have genes that code, and of course hair color is more complex than one gene, but let's just say I have a gene that codes for having blonde hair, okay? Even though some of this is not natural anymore, it is still technically blonde. It's just really, really dark blonde. All right, so I have a gene that says I have blonde hair, but actually having the blonde hair is the phenotype, which you actually see, okay? That's the difference between the two, and it's very important to understand. And then we also want to keep in mind that we have mutations, and that's going to be a permanent heritable change in genetic information, all right? A mutation of my skin cells that causes skin cancer is not going to be a heritable change in mutated genetic information. It's got to be passed on, okay? All right, so DNA is going to be a polymer. Remember, poly means many, so that's something made of repeating smaller units. If I'm going to have a beaded necklace, I was looking to see if I had anything to use in an example, that would be a polymer and the beads would be the monomers. So the monomer for a DNA is going to be a nucleotide. Okay, so we have these three parts that make a nucleotide. Okay, so we use nucleotides to make up DNA, RNA, and actually ATP is made of nucleotides as well. Okay, so for a nucleotide, you're going to have a phosphate, which is PO43 negative. Okay, basically you just need, it's a phosphate group. You just need to know it's phosphate and oxygen. And then you are going to have a sugar, which is going to be, for the case of DNA, deoxyribose here. Okay, so a 6-carbon sugar here, and a nitrogenous or nitrogen containing base. Okay, and we're going to have adenine here, and that's where we get adenine, guanine, thymine, cytosine. Those are A, T, C's, and G's that we batch up for our DNA, okay? In RNA, the sugar is going to be different, and one base may be a little bit different, but it's going to have the same structure. This is the monomer. This is one unit. We put a bunch of these together. We put a bunch of these nucleotides together with a covalent bond, all right? And that's going to make a sugar phosphate backbone of our DNA strand, okay? And it's going to attach in a repetitive pattern, and it's going to go from 5' carbon to 3' carbon. And basically what that means is how we're going to establish direction, okay? All right, so here, this is a really good structure. This here is going to be, you might see me turn to the side because I have a bigger screen next to me. It's a little easier for me to see here. This is going to be a opened up molecule of DNA, okay? And so look, you can see 5', and we're talking basically about the orientation of the bases. 5' is right here, and then 3' is right here, okay? These are what we call anti-parallel. All right, and so the strands are, so one is going to be running this way. You can see this arrow here, 5' to 3', and the other is going to be running this way, 5' to 3' this way. That is what's called anti-parallel. They're parallel, but they're going in opposite directions. Kind of like when we're on the road and we're driving a car. You're parallel, but you're running opposite sides of the car, okay? And so you've got these hydrogen bonds here between the bases. That's very important for the structure of DNA, okay? Your nucleotides are covalently bonded together. We have purines and pyrimidines, and the difference between these two you can see right here. You've got one is going to have the two sides to it, and two silic rings to it, I should say. One has the one silic ring to it. The purines are going to have shorter name, bigger size. So you have two rings here. Pyrimidines, longer name, smaller size, one ring to it, okay? And so those are going to join with what we call complementary bases. And so you're going to have a matched set, a pair, all right? Like maybe you have a pair of earrings that are complementary to each other. Or you have two puzzle pieces that fit together just right. They're complementary to each other. So you're going to have adenine that is always going to bond with thymine for DNA. And guanine will always bond with cytosine, A's with T's, C's with G's. So that is the pattern, and you can see that pattern here. We've got G bonded to C, T bonded to A, C bonded to G. That is always how they bond. It's a specific code, and that's called being a complementary base pairing, or Chargaff's rules. I'm trying to turn this little light off, but I can't. Okay. So molecules can be unzipped to be able to get to the information. I don't know what's wrong with my mouse here. It's not cooperating. Okay, so the molecules can be unzipped to get to the information that is encoded by the bases. So when we want to copy this, we have to take this helical structure and break apart these latter pieces so that we can copy those, okay? And so that's what it's talking about. This is the sugar phosphate backbone here, all right? We've got to take, I'm sorry, the rungs of the ladder here. Those are the ones we want to take off, okay? This is the sugar phosphate backbone. I said that backwards. We want to take the base pairs apart, all right? And that is there also, like I said, hydrogen bonding there. And so we said they're anti-parallel. One side of the helix is running in one direction and the other runs in the opposite direction. Let's see what else I need to cover here. I guess that's about it. So remember, RNA and DNA do have some differences. They are similar in their general properties, but the structure is different in these important ways, and I've mentioned a few. It is single-stranded, not double-stranded. Remember that double-stranded helix has to stay in the nucleus. One is too big to get out. Two, it's our $1 million copy. We don't want it to get out and get messed up, so we got to make a copy of it to keep it safe, okay? It can assume secondary and tertiary levels of complexity, which are going to lead to special forms of RNA. These are going to be tRNA and rRNA, as we already discussed. And I may move this slide, so it may not actually, I'm going to move this to where you're probably not going to already hear me talk about it. So if I hadn't talked about it yet, that's why I may move this. And so it can have those secondary and tertiary levels, which can give it those globular nature to make those ribosomal subunits. It contains uracil instead of thymine, right? Uracil replaces thymine, and ribose is going to replace the deoxyribose. It's just one carbon difference in the sugar. So replication, DNA replication is going to be semi-conservative. We're going to have about 30 different enzymes that actually take place in this. We're not looking at all of those, just about 5. And so these enzymes are going to come in. They're going to separate the strands of the existing DNA molecule, copying one strand and producing 2 complete daughter nuclei. Each daughter molecule is going to be identical to the parent in composition. Neither strand is, well, each is going to serve as a template that's going to be the original parent strand, okay? All right, so this is the list of the important molecules, particularly the top 5. Topomerase, it does supercoil and untangle, but actually, I mean, I would be familiar with it, but I would be much more worried about helicase, primase, polymerases, and ligase, okay? I know for sure those guys show up on the test. Pretty sure topomerase doesn't. So first 4 or 5, I guess that's 5. Okay, so remember that an enzyme always ends in ASC. Well, I shouldn't say always, because there's always exceptions, but for our purposes, they always end in ASC. And they tell you kind of what they do, which is really handy. So you can kind of common sense this if you just stop and think about it. Helicase, ASC tells you it's an enzyme. Heli comes from helix. All right, and so what it does is it unzips the helix. It's at the very beginning. What's the first thing we have to do if we want to copy each strand? We got to unwind the strands. And so helicase unwinds or unzips the strands, okay? Primase is going to make an RNA primer, and you'll understand what those primers are as we go on a little bit further through the process. And then we have many DNA polymerases. We've got 1 and 3 here that we're looking at. What is a polymer? Remember, I said a polymer is where you are taking monomers or subunits and putting them together like beads in a chain or in a necklace, right? You're putting them together into something larger. All right, so you have DNA polymerase 3 that is going to take those bases and bring them to add to the new DNA chain, okay? It does also work in some proofreading, okay? So it can replace mistakes in the DNA chain. We do have proofreading mechanisms, and we'll talk about this in more detail and come back around to these. DNA polymerase 1 will actually pull the primer, close the gap, and help to repair the mismatch. So polymerase 3 is going to act to find it. It's going to add new bases and then kind of proofread its work. Like if you type a letter and you go back and proofread. And then DNA polymerase 1 would almost be kind of like, let's say it this way. There's the news, a newspaper, we're going to have the writer and you're going to have the editor. DNA polymerase 3 is like the writer. They're going to add the bases and kind of proofread their work. Then DNA polymerase 1 is going to be like the editor. They're going to come through, they're going to pull away the primers because you don't need them anymore. They're going to look for gaps and repair mistakes and kind of tune things up, okay? Ligase, when you hear the word ligase, I want you to think glue. Ligase is kind of like glue, and this is going to bind together or glue in certain parts during repair. All right, so we're going to go over the process of replication. And like I said, I can't stress enough that you must watch the video over this. It's going to make so much more sense when you can actually see all these moving parts, okay? So DNA replication is outlined here in this image, and this is going to kind of show you where all these enzymes are working together. It is also showing you so many things. Like for one, we have the leading strand and the lagging strand, okay? So the leading strand is going to be copied what we call continuously. It's going to go in one direction and it's going to keep going, and it's going this way, 5' to 3', okay? At the same time, the lagging strand is going to be copied, and it's going to be copied in this direction. Now, because it's going to be copied at the same time in the other direction, it kind of jumps around, okay? And so it copies what we call Okazaki fragments. But the main point I was trying to get right here, not getting too far ahead, is that you have the leading strand and you have the lagging strand that does these little short pieces here called Okazaki fragments, okay? So we have what we call the origin of replication, okay? The origin of replication. Origin means the beginning, right? So the beginning of the process of replication. Sounds complicated, really common sense, okay? So this is a short sequence rich in adenine and thymine bases, okay? And so in biology, we like to call this the TATA box, okay? T-A-T-A, thymine, adenine, and that's what you're going to see, repeated sequences of thymine and adenine, okay? Now, our textbook doesn't use TATA box, but that's the way I learned it, and it's easier to remember. You're going to start at the TATA box, okay? The origin of replication, all right? And these are going to be held together by hydrogen bonds, okay? 2 hydrogen bonds instead of 3. So you're going to have less energy here needed to hold those 2 together. And it's going to take, actually, what I should say is it's going to take less energy to pull those apart, all right? Because guanine and cytosine are going to be bonded a little more strongly. All right, so we put a bunch of these here together so that it makes it a weak spot. And I say weak, I don't mean your DNA is weak. I just mean an area where it's easy to pull it apart, okay? Kind of like when you get a bag of something that you got to tear open. You got a bag of peanuts or pretzels, and it says tear here, and it's got that little notch. That's your TATA box, okay? You're going to pull right there and pull it all off, right? Open it up. Except in this case, it can go back together. Okay, so during replication, all right, we have those topomerases that are going to unwind the DNA helix, okay? We're going to have our helicase that's going to unzip it, all right? So your topomerases are going to kind of come in and basically unwind it a little bit, all right? And so you can kind of see that right here. Is my marker working? See that right here. So it kind of twists it, I guess is a good way to look at it. Then your helicase can come in and unzip it completely. Pull it the helix back the rest of the way apart. So those kind of work hand in hand, all right? So we're going to break those hydrogen bonds that hold the 2 DNA strands together, and then we're going to have those 2 separate strands there. And then we're going to have what we call single-stranded binding proteins, and those guys help to kind of hold them apart so they don't get back together. And you can see that right here in these little binding proteins there. They kind of hold the place so they don't come back together. I'm trying to think of an example. I don't really have a good example for that, but I don't know. If you're braiding hair, you got one hand on one side, one hand on the other. You got to keep it apart, right? I don't know how else to explain it. So anyway, DNA polymerase 3 then is going to come in and add nucleotides, okay? So if you look at these little lines, we can say these are our nucleotides, okay? And so we've got to come in and match those up with complementary base pairing, all right? So DNA polymerase 3 is going to bring the nucleotides in and add them according to those base pairing rules. A's with G's, C's with G's, okay? So DNA polymerase is going to be in the right direction for the 5' to 3' on the leading strand, okay? So this way, 5' to 3'. So it's going to go continuously, and that just means without interruption, and it's going to complete copying that strand, all right? The strand with the opposite orientation going this way, remember that's our lagging strand, okay? And it's going 3' to 5', depends on which way you want to talk about it, but 3' to 5'. From the replication fork, it's going to be 5' to 3' over here. All right, just remember they're opposite of each other. So as the fork opens up, it's going to be made, but it's going to be made kind of backwards from the previous segment, and it's going to be those little short fragments that we call the Okazaki fragments. And I believe that was after the scientist's name. That's where it gets that weird name from, okay? So you can do the copying both sides. So it's like setting your copier to copy two sides of one sheet of paper, right? It's just that it's got to do this one in little segments where it does this one all at once, okay? These Okazaki fragments this looks very small, 4 or 5. They're really 100 to like 1,000 bases long, but we got to simplify it because you couldn't see that in a picture, right? Okay, so those are going to be attached and stuck on and glued on by our DNA ligase. So that's how eukaryotes do the process. Well, what is different about bacteria? Some bacteria can add nucleotides at an amazing pace of 750 bases per second. This is because they have multiple replication forks. And so as replication continues, the new double strand will pull away from the original molecule. DNA polymerase 1 will remove those primers and start DNA synthesis and replace them with DNA. And so when they come full circle and meet together, ligase, which is our molecular glue, I was trying to say glue and ligase together, move down the lagging strand to begin linking pieces together, and then they separate the two molecules. So mistakes in replication can occur. Approximately 109 to 1010 bases can be corrected by DNA polymerase 3. And so the important part here is these multiple replication forks occurring at one time. So keep in mind that DNA does not actually perform the cell processes, but it stores the information or the directions. And then that is going to be converted into a copy, and that copy is going to be RNA, okay? And then that's going to follow the instructions to build the protein. And so this is what we call basically the central dogma, which basically means a central theme of biology. And that is the idea, and this is really important, be sure you write this down, that DNA, or let me rephrase that, genetic information flows from DNA to RNA to protein. That is the central dogma or theme of biology. You can also rephrase that as transcription, translation, and protein synthesis, making the proteins, okay? And so we got to keep these terms straight, okay? So DNA is the master code that is used to make RNA molecules. That is transcription. That occurs in the nucleus. You are transcribing, you are making a copy. What does the transcribe do? All right, and so I actually used to do this. I had an office. I listened to a little tape recorder. We would listen and we would type the information. So we're taking it from the language of DNA and putting it into the language of RNA, okay? All right, and so that is going to be our transcription. And then we are going to translate that from the language of RNA to the language of proteins. proteins. All right. So that is going to be our translation. We're translating the language. All right. And so that's going to then be used to make proteins. All right. We're going to synthesize proteins. All right. So we have exceptions to this pattern in RNA viruses, which are going to convert RNA to other types of RNA. And believe me, there are tons and tons of types of RNA. Or in the case of a retrovirus, you're going to convert the RNA to DNA. And so, like we have said before, bacteria are metabolically more complex in that they are able to be a little more adaptive and do things a little differently than we are. All right, so at the top of this figure here, we've got a circular chromosome of bacteria. DNA is the ultimate storehouse and distributor of your genetic information. On the left here, all right, we see some structures. All right, these are important. These are some really important players that you need to be sure that you know for your exam. Okay, here we have our tRNA, that is our transfer RNA. And these are not necessarily in the order that they do their work, even though all this stuff occurs simultaneously in the cell. Your transfer RNA is going to go and is going to pick up particular amino acids and bring them to this particular, this is actually the ribosomal subunit down here. It's going to bring them in and drop off those amino acids. And those amino acids are the monomers that make the protein. So it transfers the amino acid. Then it's going to leave to go and pick up another amino acid. Okay, so it's kind of recyclable. Then we have our mRNA, which is our messenger RNA. Okay, you could say mitochondrial, but that's different. That's not the one we're talking about. mRNA here is our messenger RNA, and it's the one that is going to carry the message from the nucleus. It's going to leave the nucleus because it's RNA, it's single-stranded and can come through those pores, and it's going to carry that message down to the rRNA, which is what makes the ribosomal RNA. This is what makes the ribosomal unit here. We have a ribosomal subunit. We have a large portion and a small portion that come together, okay? So those are the three main players here. DNA has got to be deciphered into a usable language, all right? So it does this by transcribing its code into that RNA molecule, okay, the messenger RNA. And then that's going to be translated into protein. So on the right here, you can see what we have are some regulatory RNAs. We have micro RNA, which is little, interfering RNA, antisense RNA, and ribose switches, okay? And these guys actually help to regulate transcription and translation. Now, you need to be focused much more on these guys on the left than we do on the ones on the right. These are more important on the left. Those are the major players. These you need to know, but these you really need to know, and you need to know what they do. By the way, if you struggle with this, I actually have a little hands-on paper template that I use in biology class that I could post for you if you wanted to work it out. I'm not sure if I have a video of me going over that or not, but you could actually walk through the different pieces. So if you want that, shoot me an email. I'll be more than happy to provide that for you, okay? So the products of transcription are going to belong into two major categories, those that are needed for translation and those that are needed for other cell functions. So we have various types of RNA, even way more than what are shown here, okay? And so we've got regulatory RNA and the small microRNA, which are also sometimes called antisense RNAs, ribose switches, and the small interfering RNA. These are going to help regulate and control gene expression in bacteria and in eukaryotes and help chromatin coil in eukaryotic cells. Okay, so remember, chromatin is going to be the loose type of spaghetti looking form of chromosomes. Chromosomes are where it's very tightly coiled. Okay, that's the main difference there. It's just describing a different state. All right. Primer RNA is going to be laid down during DNA replication for bacterial and eukaryotic cells. Ribozymes are going to be enzymes that are made of RNA and can go in and remove out those unneeded sequences from other RNA. So here's another look at these three types of RNA molecules that are really important. Here we have our ribosomal subunit, and this is the 3D globular protein here. So you have a large subunit here and a small subunit here. I'm going to lock link a video called The Inner Life of the Cell. And if you will watch that, it'll show you a lot of these processes. It's not going to be mandatory, but it is really good. And you can actually see this ribosomal subunit come together as needed, okay? You can see the whole process of protein synthesis occurring. So I really recommend you watch that one. Okay, all right. And so here is our transfer RNA. It looks kind of like a T or a boot. Remember, this is the guy that goes and picks up the amino acids, brings them to the ribosomal subunit, and drops them off to make the proteins from. Amino acids are the monomers, or the small pieces that we put together to make a protein. Okay, kind of like beads in a necklace. Each amino acid here is a monomer. This whole thing right here is the polymer, okay, of a protein. Now, this says messenger in our messenger RNA, so I'm kind of pretending this is a protein molecule, and this is amino acids here, okay, which these three codes actually do represent amino acids. Like this AUG, this codon on this messenger RNA, actually would tell tRNA, go pick up methionine and drop it off right here. Okay, so while this is actually a picture of messenger RNA, I'm using it to describe an actual protein molecule. So you could say, here is my AUG, which means methionine, and then here is my, I think that says CUC, GGG. That's going to tell the transfer RNA to go pick up a very specific amino acid and put it together, and they're going to be linked together. These are actually those 3-letter codons, these triplet codons that code for a specific amino acid. All right, so let's look at these individual steps. So transcription, remember transcription occurs where? In the nucleus. We're copying the DNA and putting it into the language of RNA. Okay. Now remember, this is different than DNA synthesis, which is where we make extra copies of DNA for things like meiosis. Okay. All right. So transcription is initiated when RNA polymerase recognizes that promoter region. Remember, that is the adenine and thymine rich region, or thymine and adenine. That's your ta-ta box. Okay. And so that's going to be in this area here. All right, so this is going to be 2 sequences of DNA right before the beginning of the gene to be transcribed. Basically a little flag that says start here, okay? It tells RNA polymerase where to bind to the DNA, okay? Then next there's a special sequence that tells RNA polymerase to begin transcription. DNA helix is going to be, the DNA helix is going to unwind. Polymerase is going to move down the DNA strand and make an RNA molecule, bringing those bases in, that is complementary to the template strand of your DNA. All right, so only one strand of DNA is copied by RNA polymerase. Next, we're going to have elongations. Well, that's the that is the process of making it longer. Okay, kind of common sense again there. And remember, we're on the leading strand. We're going 5' to 3'. mRNA is going to be put together by adding those nucleotides. All right, remember those nucleotides here are our monomers. Those are our little pieces by those complementary base pairing rules. So you're going to match it up, A's with T's and G's with C's. Okay, so it's kind of like, remember how when we were little we used to have those little decoders. All right, and so this is a code. All right, now when we're going from DNA to RNA, there is a very important difference here. Instead of having thymine, RNA has uracil. So thymine is going to be replaced with uracil, with a U. That's the only difference, okay? Well, RNA is also single-stranded, so sorry. That's the only difference, all right? So DNA is going to be rewound into its original helical form as elongation is going to continue. Then, of course, at some point, we have to stop the process. So during termination, polymerase will recognize another code that signals the mRNA strand to separate and release the transcript. mRNA is going to be about 100 to 1200 bases long, okay? And so that process is terminated. Time for another NCLEX prep practice question that applies our information here. So it says all of the following are true except for one of these for RNA. It is a single-stranded molecule existing in helical form. It contains uracil rather than thymine as a complementary base pair for adenine. Uracil does not follow base pairing rules, so the DNA code is changed. Or D, sugar is RNA and ribose rather than deoxyribose. All right, so pause this, come up with your answer. So the only incorrect answer here is C. Uracil does follow base pairing rules, and it will actually is copied from A's and T's and C's and G's. This is just going to replace the T with a U is the only difference. All right, so it is going to be C. Uracil does not follow the, I'm sorry, for a minute I was thinking, well, that's wrong, but that's what it says. That's the wrong answer. All right. So remember to really read these carefully. Sometimes it's one word, and in this case, it's to find the wrong answer. And sometimes it'll just say not. It'll have one word that's wrong. And so you got to be really careful when you read these. All right.